This narration is the summary of the International Humboldt Day lecture that I gave earlier in the week. As you know, as I talked about before, Humboldt is one of the scientists, or is the main scientist associated with the recognition of the latitudinal diversity gradient. The picture here shows it in great graphic detail. There is the highest number of species diversity in the tropics, and it decreases progressively towards the poles. Now, we've known for a long time that there are taxonomic composition differences by latitude. So you've got pictures there of tropical ecosystems versus polar ecosystems. But did that then translate to different numbers of species? And Humboldt was one of the persons that recognized it. Graphically, it's usually shown as something like this, a distribution with a peak right at the equator or 0 degrees latitude, and it works in both hemispheres with animals, plants, all across the animal, plant, various kingdoms, and it works with fossils too. Well, there's several hypotheses associated with this. Some are grouped under the whole domain of null models. There are no causal processes in this. It's just random distribution of creatures across the globe are such that there is a hard boundary at the top of the earth and the bottom of the earth. And when you draw those overlaps of where all these things are reaching their physiological optima, you just mathematically will come up with an abundance in the tropics. Okay, it's a null model. Second set of hypotheses are the most popular. They've been the most popular since Humboldt's day. The whole suite of environmental things, usually circling around the whole notion of the tropics receive more solar energy, therefore they have higher temperatures, there's higher primary productivity, and so on that way. And then you could group a whole bunch of hypotheses as interaction hypotheses. Competition creates more species creates more competition. The problem with a lot of these are that they are circular, although these types of hypotheses were most popular in Humboldt's day. Now, of course, the best ones are sort of the Goldilocks hypotheses, the integrative ones, the ones that have both. There are biological explanations and there are also area explanations. But even so, after more than a century of looking at this stuff, this area of research is still ripe. There's still a lot of people trying to find the answers. Beautiful quote from Humboldt. I'll let you read that on your own later. Let's actually dive into it a little bit. The general model looks like this, where you have the number of species increasing towards 0 degrees latitude in the center, and then decreasing the other direction, sort of a mountainous look, a hump. There are a lot of wrong models. So if you see graphs of things like this with ever-increasing diversity from one pole to another pole, or a reverse latitudinal diversity gradient with lowest diversities at the equator, or something that's constant around the globe, those are typically not viable, especially the top two. The bottom two, there are some data that suggests such things. There are anti-tropical or extratropical organisms. Just a few examples. I'll let you also see these in your textbook: plants, birds, mammals, etc., etc. And with more support than not, the pattern holds. Typically there might be little blips, like you see here, this plot of American mammals, both North and South America, has a little blip here in the south, but whatever. And again, some more exceptions. The upper one from your book, these are pine trees, and they reach a peak far from the equator. Here the equator is 0 on the extreme left of the diagram. Ichneumoniid, it's a type of wasp, a family of wasps, also more abundant in temperate zones. Just Just a few other examples of patterns. Planktic foraminifera, these are little single-celled creatures. And here diversity is contoured in the oceans. You see the contour lines and then the counts of species. And you see there's a big broad middle with 16 species across all of the tropics, all of the oceans. But you go upward, northward towards the pole or southward towards the pole, and there's this beautiful symmetric decrease from 16 to 8 to 4 to 2, 16 to 8 to 4 to 2. Beautiful pattern. Another example from the books, these are vascular plants, but here they've segregated it into 3 sort of stripes of geography across the globe. The Americas is probably the better example of the pattern, but notice here too, the peak is not at the equator, it's farther north in Central America. The stripe or examinations of diversity through Africa and Europe, very different. A huge low here reflecting the Sahara Desert, and then another peak in Europe. And then the Asia one is even more complex with multiple little humps across the bone. So there's a pattern complexity. This picture is contouring bird, Aves diversity, and notice it doesn't just parallel latitude like the planktic forams did. There's a lot of little complexity here. There's little isolated peaks of diversity, and look at this. Compare, say, diversity in Louisiana, the American South, 120, 110, 130 bird species with something much farther north, here northern Canada, 170, 160, mountainous areas 200, California 200, 210, big diversity. But if all of this is summed, you get a general picture like the one shown here. Although this is actually number of species per area, which brings up the next question. Is it just a question of area? If you were to literally measure the area of, say, the high polar regions, or not so high polar regions, but the area between 50 and 60 degrees north latitude, so a region between 2 10-degree latitude lines, there'd be 18,500 miles, but a similar latitudinal spread from 0 to 10 at the equator gives you a much larger area. So is it just a question of simple geometry? Is it the same species, but there's just more area in the tropics? Or is there greater density in the tropics? Typical density data looks like something like this. Here are birds plotted per this little quadrant, these little square equal area quadrants. And the darker the color, you see the higher density, the higher number of species per that 600,000 square kilometers. The result here obviously is that the tropics are crowded. So it's not just a question of area, it's a question of there's a reality to this diversity. The next question would be, well, how old is the latitudinal diversity gradient? We can map it today for so many things all around the globe, but how did it get there? And for this you need fossils, and as far as we can tell, we can go back at least to the start of the Phanerozoic, so the start of the Cambrian, 560 plus million years ago, and this works. And so the bottom picture, brachiopods, ammonites, forams, bivalves, a lot of the usual players in a paleontology class, and they'll have plots like this. Notice there's not data for huge portions of this, but relevant portions. So these are brachiopods of the Carboniferous. There is a peak in the tropical regions and a decline towards the poles. So it works. The next question would be, well, why the tropics? What could it be that's doing that? You could either have high speciation rates over here on the left, or you could have lower extinction rates in the tropics. As long as it would be lower than the rest of the world, They would stay in the tropics, but they would decline elsewhere. Or you could simply have the case of things are moving into the tropics. This is broadly analogous to population studies, where if you're counting the total population of a place, a country, a state, a city, you look at the birth rate, the number of new babies born, you look at the death rate, the number of persons that die, and then you also look at the immigration rate, the number of people that move in or move out. And all of these have plus and minus values to them that end up giving you that total number of people in that area. Well, we do the same thing with species, and so we have several hypotheses then. Hypothesis number one is that the tropics are a cradle. I love that metaphor. Think about that. That just means that the tropics are the place where all these new species are born. So, the tropics have more by this hypothesis just because more are born. The second hypothesis would be that there's low extinction rates. Here, the tropics are considered a museum, meaning you have species that are born here, but then they linger on, and you have lots of old species. Whereas species in the temperate zones, north and south, have a higher extinction rate. They tend to not preserve older species. And then the third hypothesis, using the same spider examples, is that the tropics are a destination. Species originate anywhere and elsewhere, but then they move to the tropics for whatever ecological reasons and accumulate there. Well, we want to test these hypotheses, and testing the first one is easy enough. You need fossils. You usually find the oldest fossil that you can of the particular group you're looking at, and that's taken to be where it originates. That's at least your best guess. And the example given here, tridacnid clams, these are the giant clams that are all over Indonesia, parts of the Indian and Pacific Ocean. But the very first oldest one is a little squirt. There it is right there, less than an inch wide, found in Oman. So still in the tropics. So a tropical origin for this. So that's a test, and it verifies then a tropical origin. So you do that for many groups. And these diagrams show that you can look at clams for different origin times, or forams or nanoplankton, and they put little checks by it. Check. You're testing that first hypothesis. You are verifying that it is possibly the case. Radiolarians, which are weird little siliceous zooplankton, do not have a peak of origin in the tropics. Instead, it's much farther south, and so they would not be the case. So what you would do to test this is look at your group of interest, other groups of interest, and literally just count the number of things. And in fact, the tropics usually are a cradle. Testing the second hypothesis, whether the tropics are a museum, requires data on the extinction rate. And here again, you need paleontological data. And these are forams, those cute little planktic creatures again. And here they've segregated this mostly northern hemisphere data into tropical origins, transitional origins, temperate origins, and extinction rates along with it. They've graphed up extinction rates, and for the museum hypothesis to be true, the red line should be entirely below the magenta and blue lines. You see that for large sections of time, look at the last, say, 6 million years, that is true. The extinction rates in the tropics are lower. But there are other time periods, look between 12 and 16 million years, where the tropics have had pulses where they have greater extinction rates. So forams in general kind of verify this, but not much. If you look at other groups, bivalves, well, here extinction rates over the past 16 million years show that the tropical areas have much lower extinction rates. So some groups will show much stronger support for hypothesis number 2. Testing the third hypothesis, whether the tropics are a destination, again, of course, requires paleontological data, but we run into the issue of a preservational bias. The tropics have typically poor preservation. A lot more species there, but because it's the tropics, with all of that extra precipitation, extra intense light intensity, much deeper, thicker developments of soils, and soils like laterites that typically are destructive for fossils, we don't have a big fossil record. Compare that to non-tropical fossils, and here you have museum drawers, we have a lot. So this one's hard to test because of this preservation bias. So somebody flipped the question and asked it the other way. Are there species that originate in the tropics that migrate outward to other latitudes and climates? This way you just have to find the one fossil of your group in the tropics somewhere that is its origin point, and then if you find it away from the tropics, you're gathering data for answering that question. And this graph is a little bit harder to understand, but if you look at it, everything to the left of the green line is deemed tropical, that has originated in the tropics but not spread out of the tropics. Look at the areas, the bars to the right of the diagram. These are things that have originated in the tropics but then migrated out of it. And as long as these bars are higher, meaning they're greater numbers and more value, you are showing that there is this definite outward migration to other latitudes. Typically, you need to use taxa with good preservation. So our best data for this are from mollusks, clams and snails, but other things are starting to show this as well. You can change this or look at different ways, different times of origin. So one of these graphs, the one on the left, looks at bivalve originating relatively recently, between, say, about 2 and 5 million years ago. And there's that pattern. More of these things migrate out of the tropics to the non-tropics. You can look at older bivalve originations between 5 or so and 23 million years ago, and again, there's more of a migration outward. So the pattern holds. We say then that the tropics can be a biodiversity engine. There are lots of things that originate in the tropics, but then they migrate outward everywhere else. So, depending on the organism, and depending upon the time, and depending upon the preservation, you can have evidence that organisms are all three of these things. The tropics may be cradles, museums, or destinations, and different relative importance of each. You need to test it. And that's where we go back to Humboldt, where he regularly encouraged the more collection of fossils and more testing of hypotheses. Incidentally, we just passed the 151st anniversary of his birthday. All the more reason to celebrate our International Humboldt Day.